Now see, let us go with the basic. First, understand what will happen when the light meets the matter. Either it can be reflected or transmitted or absorbed. Pigments are the substance which will absorb the light. Okay. Now, different pigments are there. We have studied already in the previous video, chlorophyll and carotenoids. They will absorb light of different wavelength. So ability of a pigment to absorb various wavelengths can be measured by the instrument called spectrophotometer. So why do we want to know the absorption spectrum? Which light are they absorbing? Because it will give you the clue of the effectiveness of different wavelengths for driving the process known as photosynthesis. Means they will give you the clue which light is essential for photosynthesis, because light can perform work in chloroplast only if it is absorbed. Clear, beta? So you can see here this, first of all, understand this is 400 to 700 is your PAR. PAR range from 400 to 700 nanometer. And check the diagram. This is chlorophyll A. Okay. And here check, this is chlorophyll A. So first, in the blue region, chlorophyll A has started the peak, but peak is made by chlorophyll B. Clear, beta? Chlorophyll B has made the highest peak. So first A and then B is doing the work in the violet blue region. And in the red region, this is B and this is A. So first started by B, but peak is made by the A. So what does it suggest? If you see only for chlorophyll A, you will say it is working best in the violet blue and red light, because they are the one which is the best absorbed, and green light is least effective. If you see, this light is least effective. And this is confirmed by action spectrum also. So action spectrum, if you see, first performed by T.W. Engelmann, what he has done, he has used green algae cladophora, and he illuminated this green algae by passing the white light through the prism. So different components of the white lights are created, and to check where the working is going on, he has used aerobic bacteria. The bacteria were generally used to know where oxygen evolution is more, because we'll be studying further if photolysis of water is done. So he just want to know bacteria is accumulating in which region, and he found that bacteria mainly accumulate in the region of blue and red light. Means absorption spectrum is roughly meeting with the action spectrum. But see the second diagram, analyze it. We will see that the wavelength at which there is maximum absorption is blue and red region. Here only the highest rate of photosynthesis is also going on. So it means chlorophyll A is the chief pigment associated for photosynthesis. So if we study the graph, all three graphs together, in the previous slides I have shown you separately. Now this is for absorption spectrum, this is for action spectrum, and this we are doing the comparison. So when we study, we are finding that most of the photosynthesis take place in blue light and in red light. But some photosynthesis takes place at other wavelength also. How does it happen? Though we know that chlorophyll is the major pigment responsible for trapping the light, but other thylakoid pigments are also like chlorophyll B, xanthophylls, carotenoids, and they together are known as accessory pigments. They will absorb the light and transfer the energy to chlorophyll A. So they are actually working what? They are allowing the wide range of photosynthesis to occur, because they are able to absorb some other wavelength also, which chlorophyll A is not able to absorb. Second, they can prevent the chlorophyll A from photooxidative damage also. So based on this, the topic which we discussed, absorption spectrum and action spectrum, few questions are there. Try to solve it. See, so which is the most abundant pigment present in this? So answer will be chlorophyll A. What does accessory pigment include? Accessory pigment we have seen, it is chlorophyll B and carotenoids. So answer will be this only. You can't take chlorophyll A, because chlorophyll A is your chief pigment, means it is a primary pigment. Accessory pigments, what they will do? They will pass the energy to chlorophyll A. Is it clear? They are not passing to primary acceptor. This is wrong. They are not passing to primary acceptor. Photolysis of water is not done by accessory pigment. It is done by the chlorophyll A, which is present in the reaction center. What is the advantage of accessory pigment? Advantage is that they will protect the chlorophyll A from photooxidative damage. I told you they will not help in photolysis. They are not enabling narrow range. They are enabling the broad range. Clear? And obviously, you cannot take A option. The answer is your B. So I hope so this video has helped you to understand what is absorption spectrum and action spectrum. Thanks for watching.